All right, y'all, this is one of those topics that can get overwhelming real quickly, but we're not going to do that today. So grab a snack and let's talk about cellular respiration. Hi, biology friends. I'm Sarah and I'm your other biology professor. And today we're going to talk about cellular respiration in eukaryotes, aka aerobic respiration. Now, this stuff can get complicated in a hurry, but we're going to keep it simple for now, because you can't really learn and understand the details if you don't have a good grasp on the basics. So let's go. So let's start out with a little bit of cellular geography. So we've got the mitochondrion here. The mitochondrion is a complex organelle and it's got a double membrane, so two membranes. That outer membrane separates the inner workings of our mitochondrion from the cytoplasm. That is separated from the inner membrane by the conveniently named intermembrane space. That inner mitochondrial membrane folds in on itself over and over, making these compartments called cristae in that deep interior of the mitochondrion, which is called the mitochondrial matrix. Those folds are important because it increases the surface area of that inner mitochondrial membrane. And some really important stuff happens there, so we want a lot of inner mitochondrial membrane. But important stuff happens outside of the mitochondrion as well. So let's take a look at glycolysis. This is a process that happens in the cytoplasm, and it's the part of this process that prokaryotes can do as well. Glycolysis, literally sugar breaking. We're going to take our starting material, a 6-carbon sugar, and we're going to break it into 2 3-carbon molecules of something called pyruvate. This process is also going to generate a little bit of ATP. And while it's not the focus of this, I'll keep a little table down with all of our products so you can keep track of what's being made at each stage as well. Glycolysis is important because it's going to allow for at least a little bit of ATP production, and we're going to strip some electrons off of that former 6-carbon sugar and pass them off to our electron carriers. You're going to see a lot of that. Next, in a reaction that seems to have a million names, we're going to modify those little pyruvate molecules and get them ready for the next big step. So our pyruvates are going to get a little bit of assistance passing through the outer and then through the inner mitochondrial membrane and into the matrix. And there, we're going to turn them into a compound called acetyl-CoA in a process that is, again, going to give some electrons to electron carriers. And we're going to also wave goodbye to some carbon. Whew, all right, and then things get a little bit wild, right? So still inside the matrix, we are going to start the citric acid cycle. And in the citric acid cycle, we are just going to keep popping off electrons, giving them to electron carriers, and popping off carbons until there is pretty much nothing left of our original sugar molecule. We're going to really load up the electron carriers. We did a little bit of this in previous stages as well, but this is when we get real serious about it. Because at the electron transport chain, those electron carriers finally get to fulfill their little molecular destinies and carry and drop off those electrons at the electron transport chain. Those electrons are going to get passed around to more and more electronegative proteins in the chain, finally, at the end, getting picked up by oxygen. The energy that gets released during all of this electron passing is used to power proton pumps. These are going to pump protons out of the matrix and into the inner membrane space. So what we're doing is we're generating a high concentration of protons in the inner membrane space, a lower concentration of protons in the mitochondrial matrix. Remember diffusion? We've got a concentration gradient. So what are we going to see happening with those protons? They're going to diffuse back into the matrix. Now, they can't go straight through the inner mitochondrial membrane. So how are they going to do it? They're going to go through a protein called ATP synthase, and that's going to power the production of ATP. All right, so let's recap this simplified framework of aerobic respiration. We're going to start with our 6-carbon sugar out in the cytoplasm. We're going to break it in half, do a little modifications on it, shuttle it through the citric acid cycle. Along the way, we are pulling electrons off of it, giving them to our electron carriers so that they can take them to the electron transport chain, generate a proton gradient, and that proton gradient is going to be used to make a bunch of ATP. So if you can work with that simplified framework and kind of get the big picture sense of what's going on, you can start to layer on whatever specific details of the process your professor needs you to know. All right, if you feel like you learned something, drop me a like so other students can learn a little bit as well. Subscribe for even more biology videos, more content on my website as well, and I will see you next time for more biology.